Hi, dear students. Today I am going to talk about culture media used in the microbiological laboratory. Let's start with the definition of culture media and culture. A microorganism grown in a media is called culture. The culture media consists of chemicals which support the growth of culture or microorganism. Microbes can use these nutrients of the culture media as their food. There are certain characteristics for the culture media. It should neither be acidic or alkaline. It should contain all kinds of nutrients in suitable amount, and it must be sterilized before its use. Sterilization means free from microorganism. The media introduced by Robert Koch. Meat infusion broth is the first introduced media. Petri dishes are used for preparing the Petri plate. Petri plate means Petri dish containing media. It is introduced by Richard Petri. Next, we can discuss the uses of the agar or the components of the agar-agar. Agar is a solidifying agent and it is a sulfated polymer composed of mainly D-galactose, 3,6-anhydro-L-galactose and D-glucuronic acid. Agar-agar is extracted from the red algae. It is used as a solidifying agent because it melts at about 90 degrees Celsius, but once melted, it doesn't harden until it reaches about 45 degrees Celsius. Thus, after being melted in water, it can be cooled to a temperature that is tolerated by human hands and microorganisms. So, it is an excellent hardening agent since most microorganisms cannot degrade it. Media classification. Media can be classified in many ways. Based on the state, we can classify the media as solid media, liquid media, and semi-solid media. It is a type of classification. Then another type of classification as simple media, complex media, synthetic or defined media, semi-defined media, and special media. Special medias include enriched media, enrichment media, indicator or differential media, selective media, sugar media, and transport media. Then the third type of classification is based on oxygen requirement of the microorganism. Based on it, media is classified into aerobic media and anaerobic media. We can discuss each and every media with example. Solid medium. When 5 to 7% agar-agar or 10 to 20% gelatin is added to the liquid broth, then it becomes solidified. Such media are used for making agar slant or slopes, agar stab or Petri plate, etc. Example for the solid media: nutrient agar, eosin methylene blue agar, etc. Second type of the media, liquid media or broth. In this, no agar is added or used while preparing the medium. After inoculation and later incubation, the growth of cells become visible as turbid solution, and the turbidity can be measured by calorimeter. Example for the liquid media are nutrient broth, peptone broth, etc. Third type, semi-synthetic or undefined media. Example for the semi-synthetic media is potato dextrose agar. It is a media where the exact chemical composition is unknown. Okay. Selective media. Selective media favor the growth of a particular microorganism. If the inhibiting substance is added to a solid medium, then it enables a greater number of the required bacteria than the other bacteria. Example for the selective media is deoxycholate citrate medium of dysentery bacilli. Some media contains bile salt and dyes. Dyes like basic fuchsin and crystal violet. This will favor the growth of the gram-negative bacteria by inhibiting the growth of gram-positive. These dyes have no effect on the growth of gram-negative bacteria. Okay, so the inhibiting substance, that is the dyes and bile salt, inhibit the growth of gram-positive. Okay. Another examples for the selective media are endo agar, eosin methylene blue agar or EMB, and McConaughey agar. These three agar media used to detect the presence of E. coli in the water sample. These three media contain dyes which suppress the growth of gram-positive bacteria. So it allows the detection of presence of E. coli or the coliforms in the water. Another example for the selective media is CMC agar. The medium containing cellulose is effective in the isolation of cellulose digesting bacteria. Okay, CMC agar contains cellulose that is effective in the isolation of cellulose digesting bacteria. This is all about selective media. Differential media. It is a media used to distinguish different types of bacteria, and it allows the identification of microorganisms based on the biological properties. Example for the differential media is McConaughey media. McConaughey medium consists of peptone, lactose, agar, neutral red, and taurocolate. It shows lactose fermenters as pink colonies, while the non-lactose fermenters as colorless or pale. So the lactose fermenting microorganisms appear as pink colored colonies in the McConaughey medium. Okay. McConaughey media is an indicator media. Another example for the differential media is the blood agar. Blood agar used to distinguish hemolytic and non-hemolytic bacteria. It is a type of enriched media and it is an indicator media. Next type of media, selective differential media. This type of media have the property of both selective media and differential media. Okay. This media favor the growth of a particular microorganism and is used to differentiate a bacteria from others. That means this media exhibit both as selective and as differential media. Example, McConaughey agar. In the McConaughey agar, the selective components are bile salt and crystal violet. These components inhibit the growth of the gram-positive bacteria and allows the growth of the gram-negative bacteria. And the differential media components are lactose, neutral red. So these components can differentiate gram-negative bacteria based on lactose fermentation. Next type of media, indicator media. This media contain an indicator which changes the color when a bacteria grows in that media. Example, sulfite in Wilson and Blair medium. Sulfite reduced to sulfide in the presence of growth of Salmonella typhi. So the Salmonella typhi shows black metallic sheen in this media. Another example for the indicator media is potassium telluride in MacLeod's media. Potassium telluride reduces to metallic tellurium by the growth of Corynebacterium diphtheriae. By the reduction process, the Corynebacterium diphtheriae bacilli produces black colonies. Eighth type of media is transport media. Some organisms cannot survive the time taken for transporting the specimen to the lab. In such case, a special media used for transporting the specimen. Okay, example, Stodd's media. Stodd's media contains a reducing agent, charcoal, and glycerol saline. Glycerol saline provide the growth of enteric bacilli, and the function of charcoal is to neutralize the bacterial inhibitors. Then a reducing agent used to prevent the oxidation. So the transport media used to transport delicate organism. Example for the delicate organism is gonococci. Next one, Enriched media. Enriched media used for the growth of fastidious microorganism. Fastidious microorganism means some organisms require more nutrients for their growth. These are fortified media. That is, the media contain either blood, serum, or egg. Example: blood agar, chocolate agar, egg media, etc. Another type of the media is enrichment media. In material containing more than one bacterium, then the bacterium to be isolated is overgrown by the unwanted bacteria. That is, pathogenic bacteria overgrown by the non-pathogenic bacteria. Such condition may create some problems in isolation process. Example: Salmonella typhi overgrown by Escherichia coli in cultures from fecus. In such cases, a substance that have a stimulatory effect on pathogenic bacteria and inhibitory effect on non-pathogenic bacteria are added to the media. So, that will promote the growth of the wanted bacteria and suppress the growth of the unwanted bacteria. Another example for the enrichment media is tetrathionate broth that inhibit the growth of coliforms and allows the growth of Salmonella typhi and Salmonella paratyphi. 11th type of media is simple media, otherwise called basal media. The best example for the simple media is nutrient broth. Nutrient broth consists of peptone, beef extract, sodium chloride, and distilled water. Nutrient broth can be converted to nutrient agar by adding 2% agar. If the agar concentration is reduced to 0.2 to 0.5%, then semi-solid or sloppy agar is obtained. Okay, semi-solid or sloppy agar used to isolate motile organism. High concentration of agar prevent spreading or swarming by the organism. Next type of media, complex media. It is a media used to bring out certain characteristic or providing special nutrients required for the growth of bacteria. That means the complex medium contains some ingredients for the special purpose. We can define the complex medium as media that contains some ingredients of unknown chemical composition. Such media are very useful. If the nutritional requirement of a bacteria are unknown, then we can use complex media instead of defined media. Complex media contain undefined components like peptone, meat extract, beef extract, etc. Peptone are the protein hydrolysate prepared by proteolytic digestion of meat, casein, soya meal, gelatin, and other protein sources. It serves as a source of carbon, nitrogen, and energy. Yeast extract is the extract of brewer's yeast. It is an excellent source of vitamin B, nitrogen, carbon, etc. And the beef extract is the extract of lean beef. It contains organic acid, amino acid, peptide, vitamins, minerals. Examples for the complex media are tryptic soya broth, McConaughey agar, etc. Synthetic or chemically defined media. This media prepared from pure chemical substance. That is, the exact composition of the medium is known, and it is used for the study of various metabolic requirements. Example, Zapac-Dox media. Then we can move to the last type of media, that is anaerobic media. It is a media used to grow anaerobic organism. Example, Robertson cooked meat media. That's all about types of media. I think you got an idea about culture media, agar, then different types of media, media contents, uses, examples, etc. Thank you all for watching this video.